So thank you all for joining, and thank you, Roman, for accepting to present. So Roman is a lecturer at the University of Surrey, and is specialist of multi-agent modeling. Especially, I was interested in his work because I came across the Biodynamo platform, software, which is a way to simulate multi-agents. And so he will tell us about his work in biological modeling today. Thank you very much, Roman.
Thank you, David. Can you see the slide?
Yes.
Perfect. Right. So first of all, thanks for having me. Today, I would like to talk about some computational approaches to capture biological systems and dynamics of systems, including self-organization. But then towards the end of the talk, I will also look into another biological system and another approach that can capture various aspects of this biological system. So this talk is meant to be a showcase of different approaches and some example applications of those computational approaches. So on a fundamental basis, we can differentiate two extreme versions or two ends of a spectrum of models. On one end, we have model-based approaches where we create a model that somehow incorporates existing knowledge about the biological system, for instance. So we create a computation model that captures, for example, dynamics of gene regulation or some kind of migration of cells or bacteria based on some kind of equations that we have previously already inferred to capture quite well that system. So we have already some kind of knowledge that we incorporate into this computational model in order to mechanistically capture the dynamics. And then on the other hand we have data-driven approaches where we have little mechanistic insight potentially, but we have a lot of data, and so, for example, large language models are examples of such a data-driven approach where we have a lot of written text and so on, which then fed into a neural network that then just creates a data-driven model of the underlying input. And then there are, of course, also mixtures of those. So it's not like it has to be one or the other. One can also have hybrid models, and they have their advantages and disadvantages, of course. And I want to focus in the first part of my talk on a model-based approach, a mechanistic approach, and also highlight some of its advantages. And then later on, I will also elaborate a bit more on a more data-driven approach. So, from a model-based approach, one example is agent-based modeling, where we model interactions between biological elements among each other, but also with a physical external environment. And from a biological perspective, we want to then model how do the biological rules that are encoded in the genes and that are expressed via gene expression, how do they interact with the external environment of cells? So we have, for example, mechanical interactions between cells that also influence how cells behave. We have, for example, chemicals, extracellular substances that diffuse in space and that act as guidance cues, for instance, or they can nurture cells. And there is, for example, also electrical activity. For example, if we speak about neural networks or neural tissues. And it is this interaction that we want to capture with a computational agent-based model of a biological system if you want to model, for example, self-organization and development of biological systems. And importantly, in an agent-based system, as I said, we have agents, and that's why it's called agent-based modeling, that are autonomous. And so we model their behaviors as autonomous agents with their local environment only. And so there's only local information exchange. There's not like a global supervisor, as you can see here, that tells the individual agents what they should do, but they follow their internal rules and they can sense and interact with the immediate local environment. Now, what kind of biological behaviors are we speaking of? So until now, I've worked with agent-based models for like more than 15 years now. We have used those 7 fundamental basic behaviors, and until now, those have been sufficient to model any kind of biological system that we wanted to model. So what are those behaviors? Well, one behavior would be for an agent or a cell body, for example, to change its physical properties. For example, it can increase in size, it can shrink, it can change maybe its adhesive properties and so on. So this is a rather generic behavior, but it is still I think a good kind of elemental behavior. Then we have the behavior of move. So cells can, for example, migrate by sensing chemicals, so they can change their location essentially. Then we have spawn, which is like a generic term to refer to cells proliferating. Like a cell can divide and give rise to 2 cells, or we can also mean by that a neurite grows out of a cell body. So if you have a neuron, for example, and we have a cell body, then you can see the extension of a neurite from that cell body as a spawning event. So it's like the birth of a new type of agent. Then we have secrete, and here we mean that cells can secrete some kind of entity. It can be like a chemical. It could also mean actually electrical activity too. So you kind of emit some kind of electrical signal. So it's something that the agent kind of emits into physical space. And then we have detect. So this will mean, for example, that the cell can sense a guidance cue, or can sense a gradient of a guidance cue. And then we have die. So cells can die. They can commit to apoptosis or necrosis, so they can vanish. And finally, bond. So this could be, for example, a physical bond between 2 cells, 2 cell bodies, or this can be also a synapse between neurons. So this is some kind of bonding mechanism. And as I said, until now, those 7 fundamental behaviors have been sufficient for us to model any biological system that we have looked at. And we have looked at a wide range of different biological systems, as you will see. I will give some examples of biological systems that we have modeled with agent-based modeling. Then from a physical perspective, what kind of mechanisms or processes do we want to model in such an agent-based model if we look at the physics? Because ultimately we want to model how the biological behaviors interact with a 3-dimensional physical space. And in this 3-dimensional physical space, we want to, for example, include the diffusion of substances, as I briefly mentioned before, or the decay of a substance. Also, we want that the boundary conditions can be customizable. So we want, for example, to state that the boundaries are closed, or they are open, or they are fixed at a certain value for a concentration, for instance, or for a temperature. Then important, as I mentioned, is that we should have some kind of mechanical interactions, both between cells, but also with the external environment or a substrate. Thermodynamics is something we have looked at. I will not go much into detail here because it's a bit beyond the scope self-organization or complex biological systems, but we have also modeled how cells behave in a thermodynamically changing environment. And fluid dynamics is something we have especially recently looked into in more detail. And then finally, radiation is something also we have modeled with agent-based modeling. So the impact of radiotherapy on cells, including cancer cells. So this sounds all quite abstract. So I want to give you an example of a biological system that we have investigated in this manner. And one example that we have looked at is the so-called layer architecture of the mammalian brain, especially the layers of the cortex. And here you can see with a Nissl stain that the brain of I think it was this was from a cat. You can clearly see that there are different layers of cells. So the cytoarchitecture is different for different layers, and we broadly summarize this as 6 layers of neocortex. And so the question that we addressed is, can we create with agent-based modeling a model that can explain how this architecture develops or self-organizes? And what is also interesting is that this is a very universal architecture, but the layers and their thicknesses and the numbers of cells in those layers changes a lot depending on what kind of species you look at. So different species have different types of layer architectures, and also it changes actually even within the same brain or same species, but in different parts of the brain. So if you look at, for example, the number of neurons in the human temporal cortex, then here you see some visualization of the number of neurons in the different layers, in the, for example, in the mouse barrel cortex or mouse frontal cortex, you have quite a different number of neurons in those different layers. And also, even if you look at it proportionally, you see quite a big difference across different species, or even in the same species, you can see differences there. And so one question that arises is, if we can create such a computer model of how the layers form, can we also make it flexible enough to capture those differences and this variation across different species and even within the same species? And to cut the story short, we did this by creating a computer model of a gene regulatory network. So how do the cellular dynamics, internal dynamics, produce differentiated cells? So these are 6 different cell types: the marginal zone, layer 6, layer 5, layer 4, layer 3, and layer 2, and they differentiate from a stem cell pool of undifferentiated progenitor cells. And we also have some apoptosis. So the A stands for apoptosis. You don't need to understand this in great detail. The important thing here is just that we created a gene regulatory network model that is biologically plausible and that produces different types of cells. And then we simulated this with agent-based modeling. Here you can see the initial stage on the left-hand side. We have an undifferentiated pool of precursor cells, and they then produce different types of cells, and also they produce them in the right order. So somewhat counterintuitively, it's not like that you have first layer 1 developing and then layer 2 and then layer 3 and then layer 4 and 5 and 6, but actually we have first the marginal zone developing, and then the deeper layers where we have layer 6 later on developing, and then actually layer 5 develops and layer 4 later on and pushes the marginal zone up, then layer 3 and 2 and 1. So it's this inside out development sequence that we reproduced in simulation. And we could also reproduce proportionally the right numbers for different parts of the brain and in different species. And so this is a nice proof of concept that we can create a mechanistic agent-based model of how the layers of the brain develop. And just to make it a bit more visual, here is a video where we simulate this. This is from the side, a view from the side. So it's a 3-dimensional simulation. But it's just for better visualization. You can see it from the side, and so you can clearly see here how the cells push up the marginal zone. And importantly, each interaction, mechanistic interaction, is here explicitly modeled with agent-based modeling. And in this case, we use the software called Cortex 3D, which is an agent-based modeling software developed in Zurich at ETH Zurich and University of Zurich Institute of Neuroinformatics. And so you can clearly see here that we have this temporal sequence reproduced in this video. So this is just a proof of concept, essentially. But what is important here to notice is also that, as I mentioned, we have a lot of interactions here. And so this is quite challenging from a computational perspective, both in terms of software, because we need to have good software for this, but we need to also have hardware, essentially we need to make the most out of the hardware that we have. We don't want this to become something that only very large companies can afford to do. Ideally, everyone who has a laptop should be able to produce some nice, realistic simulations. And so we thought about how to create a software that is very efficient. Before I get to that, this is just an example to also show that these simulations are biomedically relevant. So we could actually reproduce some neurodevelopmental diseases characteristics like polymicrogyria, where we have a very thin cortex, or in autism spectrum disorders, where the cells are more disordered. But again, this is not something that I want to go in detail. It's just to show that those simulations are also relevant from a biomedical perspective. But back again to the performance issue. So we created a consortium called Biodynamo that involves now 8 organizations. So at the very beginning, it was just CERN and Newcastle University, and then we have grown to a consortium of 8 prestigious international organizations who are involved in this collaboration. And we have created software based on various techniques. And one of the important techniques that we use are code optimization techniques. And here, for example, you see a paper where we published some code optimization methods in order to render agent-based modeling much more efficient. And actually, we improved, or we scaled up the efficiency here with those code optimization techniques, I think by about 500 times or so. So it was really a huge improvement as compared to non-optimized software. And in that case, we had simulated, I think, 32 million cells that interact with one another. And there is also diffusion of substance in this 3-dimensional space. And in this case, we have 2 different cell types. And as you can see here, over time, they kind of form those clumps, blue and orange clumps, because the blue cells secrete one particular substance that they are attracted to, and the orange cells secrete another type of substance that they are attracted to, and so that's how you get those clumps over time. This was just an example simulation to kind of benchmark the performance, and as I mentioned, we could really scale up significantly the performance by using sophisticated code optimization techniques that helped us then also create the Biodynamo agent-based modeling software. So this is a highly efficient software that we have written based on this prior work and additional new research insights. And this software is open source, so anyone can download it and run it for free. And there's also documentation on the website. So if anyone is interested, I would suggest to have a look at the website, or feel free to get in touch with me if there's any questions. But the goal was to create an agent-based modeling software that allows to model different kinds of scientific topics, both in, for example, in computational neuroscience or in cancer, or for example, biofilms. I will later on also get a little bit into biofilms, because I know that some in the audience here are interested in biofilms. So the important thing was really to create a software that is versatile enough to be able to model a wide range of different complex systems with agent-based modeling, and to do this potentially just on a normal laptop. So we can actually use high performance workstations, but we can also run BioDynamo on a normal laptop. If it's not like millions of cell elements, that's absolutely fine. And so importantly, it allows to combine those biological dynamics, as I mentioned before, and also to simulate physical processes, like in this case, for example, the diffusion of substances and the decay of substances and the production of substances by the cells. And so we can really model this tight interaction and complex interaction between biology and physics. So just to give us some examples on what we have done with BioDynamo and what we are doing, one of my PhD students, Mr. Umar Abu Bakar, is working in computational neuroscience, and he's essentially continuing this line of research with the layers, but now he's also adding neurites to this. So we are creating entire neural networks that are biologically plausible and that develop in a self-organizing manner. So as you can see here, we are simulating the development of complex neural networks that then also connect to one another in a biologically realistic manner. And then we can measure also the connectivity between neurons, and we can compare with experimental data in order to create models that are not only explanatory for experimental observations, but then also can create new kinds of predictions potentially. And so there's a lot of work that one can go into this direction, also with regards to scaling up to larger neural systems. So here you see, for example, a large neural network growing from a very simple setting, where we have, I think, on the order of millions of neurite elements. So in total, there are millions of agents, autonomous agents, that we simulate here with BioDynamo. And then we can really ask a lot of questions, like how do certain neurodevelopmental diseases arise? What kind of potential options to intervene are there realistically to improve outcomes if there's such a disease occurring, and so on. So there's a lot of also biomedically relevant research that can be done. Then just to give another example, we also work in the consortium on cancer, and in this paper that we published a few years ago by a previous PhD student of mine, Jean de Montigny, where we simulated in 3 dimensions how cancers grow, but then also how does, for example, the distribution of oxygen in 3 dimensions evolve over time, or the density of necrotic cells. So we can really create like an in silico laboratory to test different hypotheses and to compare with experimental data. And we can do this, and then we can probe it, and we can analyze it at great detail. And this kind of experiments, of course, you cannot do experimentally, right? You cannot really over time in 3 dimensions measure oxygen distribution and saturation levels and so on. But with in silico modeling and including agent-based modeling, we can create very realistic and sophisticated models in order to then also reproduce various experimental data to see if those models actually make sense. Here, for example, in the paper where we present BioDynamo, we also reproduce some experimental measurements of how cancers grow different types of cancerous systems depending on the initial conditions. So the colored sample points indicate experimental results. So here, for example, we had a different initial condition, and then we see the diameter of this in vitro cancerous system grow over time. So first it actually shrinks, but then it grows. And here you can see this for a different initial condition. And we could reproduce quite well with BioDynamo this kind of in vitro experiments and compare them with experimental data, which is another important aspect of agent-based modeling, because comparison with experimental data can be done on a multi-scale basis. So we can really model gene regulation, as I showed at the beginning, but we can also model number of cells, like how many cells are there. We can model concentrations of extracellular substances and so on. So on multiple scales and also on temporal scales, so both spatial scales and temporal scales, we can create models that then can be compared with various experimental data. So it's not like we are kind of constrained to a very particular experimental type of data, but there are various different modalities, data modalities that we can use in order to assess whether our computational models make sense. And then we can refine the computational model and create predictions, predictions that previously have not been experimentally assessed, ideally. So it's really a new type of type of experiment that is proposed by the in silico modeling, and then we can refine the computational modeling and improve our understanding of the complex system iteratively. Also relevant to cancer is a topic that a previous student of mine that I co-supervised works on, Dr. Niccolò Conio. He's now a doctor. He has been looking into lung fibrosis, and in particular radiation-induced lung fibrosis. So in cancer therapy of lung cancer, one of the big problems is that radiotherapy can lead to fibrosis, which can be very dangerous, of course. And so the question is, can we create a model, an in silico model of this system in order to optimize the radiotherapy? Because you don't want to have too much radiation, because if you have too much radiation, you kill a lot of healthy cells, and you can get this lung fibrosis complications. But you also don't want to have too little radiation, of course, as you then won't affect the cancer cells significantly. And so what Niccolò did in several papers is he created a very biologically realistic and very sophisticated computation model of lung cancer in 3 dimensions. So he modeled the alveoli of the lung. He modeled even intracellular signaling and interactions with extracellular, like here, for example, the extracellular matrix, the interactions between extracellular and intracellular dynamics, and he then also interfaced BioDynamo. So the agent-based model was done in BioDynamo. He interfaced that with a Monte Carlo model that simulates the impact of radiation on single cells. And by using this interfacing, he could extrapolate, so to say, the in silico modeling to entire tissues and entire alveoli of the lung. And he could then create models of the radiation-induced lung fibrosis, or RILF means radiation-induced lung fibrosis. So this is something we want to minimize, of course, as long as we have an impact on the cancer. And so this is the overall process, or the overall kind of in silico approach. It's not only agent-based modeling, but one of the big advantages of agent-based modeling is also that it allows to be combined with other models, like in this case Monte Carlo modeling, And then we created this sophisticated approach in order to essentially look at different dosages of the radiotherapy and treatment protocols, and how it affects real severity. And as you can see here, for instance, the simulation experimental results, both in solid and dashed lines, agreed very well. In last year in Nature Communications. We got nominated for one of the breakthroughs of the year in physics for 2024 for this, because it allows to really create a physically realistic but also biologically realistic simulation of radiotherapy in lung cancer, in order to help improve outcomes, particularly due to complications from lung fibrosis. And so this is also, I think, nice to see or to highlight that these kind of methods are highly interdisciplinary. And so we are within Biodynamo very much collaborating with experts from different fields. There's not one person that knows everything about radiotherapy and cancer and agent-based modeling and code optimization and so on, as you can, as you know. And so we are highly interdisciplinary collaborators, and this is just due to the fact of the problem. Then something that, as I know, is quite interesting for some in the audience here, is that we also look into biofilms, and in particular into how biofilms interact with the substrate that they grow on, and different conditions, environmental conditions. And my student Ryan Burns is looking into this, and he created some very nice way to import into Biodynamo meshes of substrates, very complex meshes that reflect, for example, the PVC of PVC catheters. Like in catheters, one can also have the problem of biofilm formation, and this, of course, is a medically relevant problem. And so what he did is he can actually import into Biodynamo some surface meshes that reflect the surfaces of PVC catheters, and as you can see here, these are really complex surfaces. So we can go beyond those boundary conditions, which are simple, smooth boundaries, but we can now have very realistic boundaries, and we can model various physical processes, and also the interactions between bacteria and those substrates. And so one topic that Ryan is looking into, or has been looking into, is the attachment of biofilms to the substrate, and how this depends on the form and the shape of the substrate, or the surface of the substrate. And here you can see how those biofilms in simulation attach while there is a flow dragging them essentially across this surface. And there are two different types of agents here. There are the EPS that kind of hold together biofilms, and then the bacteria, and you can see here that those really attach to the substrate in this simulation. And there's, of course, also a lot of potential here then to compare with experimental data in order to optimize, for example, the substrate. Then we also simulate complex fluid dynamics in interaction with those biofilms and with the substrate. So there's a lot of things that come together here, right? So we simulate biology in the form of bacterial behaviors. We simulate complex fluid dynamics by interfacing BioDynamo with OpenFOAM, which is a library to simulate such processes. And then we also simulate the impact of the substrate and the mechanical interactions. And you can see here that in this case, there is some clumping of cells, but there is also individual cells that are kind of dragged by this flow. And so there's a lot of complex things happening here that we can now investigate in simulation by using BioDynamo. This is another example of such a detachment induced by flow. In this case, you can see much more clumping or sloughing as it's also named, where many cells are detached due to the influence of this flow. And again, this is computationally a very demanding task, as you can imagine, because all of those cells are interacting with their immediate neighbors. And so one needs to have the proper software in order to efficiently simulate this. Otherwise, it becomes extremely or too demanding to really do this with reasonable computer resources. I mean, if you're not like Google that has like thousands or hundred thousands of servers that can do this, we really needed to think about how to create efficient code to do this. Also then later on to look at parameter sensitivity analysis, right? We want to then understand how do different parameters affect the outcomes of these simulations. How do sometimes if we find out that some parameters are very sensitive, so small changes may have a big impact, we need to find or identify better experimental data to set or determine those parameters, and that takes a lot of simulation runs to really assess the impact of parameters on the model outcome. Right. So these were some examples that I've shown now from neuroscience, from cancer research, from biofilms. As I mentioned, BioDynamo is open source. It's on GitHub. You can download it for free. We are an open source collaboration. We are not a for-profit organization, so we welcome contributions very much. We welcome collaboration. So have a look at this website if you're interested in topics relevant to this. And there are also documentation like how to get started. We regularly do also schools like tutorials and training schools to help people, including experimental people, learn about how to use BioDynamo. So have a look at this website. As I mentioned, we do these training schools, and the one is upcoming very soon in Zurich on the 6th of July. Coincidentally, this is my birth town and this is my birth date. So this must be a sign, I guess. So if you're around and interested, please come along to this European Society for Biomechanics conference or congress, and then we will have a preschool, or pre-course, sorry, on BioDynamo, where we will go a bit in more detail into how to make use of BioDynamo, how to look into those demos and try them out. So that was one approach, one age-based modeling approach, a mechanistic approach, which has a lot of advantages. But of course, as I mentioned, there are also challenges with regards to, for example, to parameters. One needs to know parameters, or at least the range of plausible parameters. If we follow a more data-driven approach, then an advantage is that we need to rely on less predetermined parameters, because everything comes essentially from the data. But a disadvantage there is, of course, that we need usually a lot of data, except we use some very sophisticated computational or machine learning methods. And this is what we are doing here in this project here. where we look into retinal fundus images in order to create biomarkers for certain conditions, like neurological conditions. So what is a retinal fundus? So the retina is a neural tissue at the rear of the eye, and it can be imaged, or one can create photos of the retinal fundus very easily. You don't need to be a doctor to be able to create a retinal fundus image. Actually, you can go to an optician, or if you create if you buy some new glasses, one can actually quite easily create relevant images, and it's also very cost efficient. So it's something that can be done. I mean, it just doesn't cost almost nothing. It's a few cents or so, and also it's very quick. So, for example, if you compare to MRI-based imaging, MRI takes maybe an hour or so, and it's very difficult for some people to do who maybe are claustrophobic or so. And so one of the big advantages of retinal fundus images is that it's something that can be done very quickly and non-invasively. So you don't need to take in some radioactive tracer or something like that, like in certain PET scanning based diagnostic measures. But here you can really do this all externally, and also and due to those advantages, it also has high patient compliance. So it's a large number of patients can do this. Usually there's no major issues here, and because it's so quick, and because it's non-invasive, and because it has high patient compliance, it's something that can be done over time. So you can really assess many times. You could do this every week or every month, and you can look at changes. And due to those advantages, of course, people have been looking into the diagnosis of certain diseases based on the retina. And for example, here in this paper here, people have really achieved very good success rates to, for example, assess cardiovascular risk and other issues. But one of the problems is that these are usually not explainable. So from a medical standpoint, if you have a method that gives you some kind of diagnosis, you want to understand why this diagnosis came up, right? That's also the case, for example, in financial problems, for example, right? You don't want to make or invest millions or billions into something if you don't know why the AI tells you to do that. And the same is true in medicine. And so this is one of the challenges currently with regards to the diagnosis of retinal based on retinal images, is that usually this kind of deep learning methods are black box approaches. And so it's very difficult to really understand, not only for the patient, but also for the doctor, why a certain conclusion came out of that. And so this is something we are tackling in collaboration with colleagues at Surrey, but also at other universities and in Newcastle and so on. And so how do we do this? So one first step is to the processing of the retinal fundus image, where we can segment the vessels. So one can extract here and infer where the vessels are. And then we can classify arteries and veins. And then we can look at certain characteristics of those arteries and veins. And we have done that in the past, and we published this, and we can actually create features from, or extract features from those images that are explainable. And then we can also create models based on those features. And just to go a bit more into detail here, so how is this done? So when we have the retinal fundus image, we can then have those construct those zones A, B, and C, and then for different zones we can extract various features. And this is all done, of course, automatically. So it's not like we need to manually designate where those zones are, but this is all done automatically. And then we can extract those features, and we can use some kind of machine learning methods in order to create classifiers that diagnose conditions like neurological conditions. And we have looked into Alzheimer's disease, Parkinson's disease, and neurovascular dementia with very good performance from the UK Biobank, with data from the UK Biobank. So here you see some results on the performance. So we used some methods called PyGol or NumLoc, which are methods from inductive logic programming. So inductive logic programming is an AI method that is not a black box method. So it's a different branch of AI. And as you can see here, for the different types of tasks, we could achieve very good diagnosis or accuracy for Alzheimer's detection, Parkinson's detection, as well as neurovascular dementia detection with PyGol, with this ILP, inductive logic programming method. And the nice thing is also that this method allows us to create a hypothesis. So we feed it data, we extract the features, and we train PyGol, and then it tells us certain types of hypotheses that seem to be true, and for which, of course, there's a lot of evidence that it is true. And it tells us, for example, if certain conditions on those features are given, like for example, if the tortuosity artery value is higher than this value, or if the tortuosity vein value is lower than this value, and so on. If those are given, then the classifier says this person has Alzheimer's disease, or it's at high risk of having Alzheimer's disease. And so we can create hypotheses that then can be experimentally also assessed, and then we can use that in order to better understand also the progression of the disease, right? In order to really create mechanistic insights into how the retina is affected from Alzheimer's disease. The retina is part of the brain, is a part of the central nervous system, and so it makes sense that the retina is affected by Alzheimer's disease because it is connected via the projections from the retinal ganglion cells. But to really create quantifiable predictions is something else, and that's what we are able to do. And we have written a manuscript which has been recently accepted at the IEEE conference on AI, and we had presented this just 2 weeks ago in Santa Clara at the Bay Area, and so this will be soon published. And so to summarize, I would like to highlight that overall, we can see kind of two extremes of a spectrum of computational approaches. One is mechanistic, model-based, and the other one is data-driven. And agent-based is a mechanistic approach, and it can serve as a powerful framework to really simulate self-organization and other types of biological tissue dynamics, including formation of biofilms and detachment. And then we have created a software which is highly efficient and which is available as open source and which can be interfaced with other computational tools. And within my lab and with our collaborators, we also work on data-driven and explainable methods to capture such complex biological systems like images of the retina, and then we can create even there some potential mechanistic hypotheses that then can be validated with new experimental data. So if you're interested in any of those topics, please don't hesitate to get in touch. We are very interested to work on different problems, but ideally with those approaches, of course. And so I would like to thank you for your attention. And as I said, if you have the opportunity, please come along to the ESB Congress in Zurich in July. And if you have any questions, then I'm happy to give it a try and address them. Thank you.
Thank you very much. Very nice talk. I think it shows greatly the versatility of agent-based modeling and biodynamo in particular. Are there questions in the audience? I think yes, Lennon has a question. We can't hear you, Lennon.
Now, can you hear me?
Yes.
Okay. First of all, thank you for the presentation. Really nice, and really nice to see how much applicability you have, like in different kind of cells and in different kind of organisms too. I am more interested in the biofilm. Was really nice presentation. I have actually 3 questions. The first one, I saw when you showed the biofilm, you have 2 colors in the cells. Is different species, is different cells, or what is the difference between them?
Yeah, so the two colors indicate, so one color indicates the bacteria, and the other color indicates the EPS, which essentially is secreted by the bacteria, but which makes them stick together.
Okay. And do you, when you also, it's a general question, when you enter like the characteristic for the cell, for the bacteria or either for the cancer cell, you have like a set of setup that you need to configure what kind of cells, how long they stick there, or what what they produce?
Yes. So that's a very important part of this research is about the calibration of the parameters. And actually, we published a paper. I should have referenced it, but we published recently a paper on calibration, model calibration in agent-based modeling, and so there's a lot to be said there. I mean, it's actually a long review paper. So I would maybe, if you go to my Google Scholar and just check, I think it was published a few months ago, and so, well, one important source of knowledge to determine those parameters is of course experimental data, and we try to, as much as possible, determine those parameters with experimental data, but often the parameters are under constraints. So often we have a range of biologically realistic parameters, and we have to choose one. So our approach has been, as much as possible, determine the parameters based on experimental data, and if not completely constrained, select something that is not implausible, so that makes sense, and see what the results are. If the results are in agreement with the experimental results overall, because we simulate the dynamics, right? So we can assess then after some time, what does this lead to? What does this system converge to? And if those are sensible, then we stick to those parameters and we say, look, this is not a precise parameter setting that 100% surely has been determined based on experiments, but it shows that in those ranges we get reasonable experiments that make sense and that are biologically realistic. But as I said, this is an interaction with experimental researchers. So at this stage, we are not able to create an in silico model that 100% surely captures all of the things of the biological experiments, but we need to interact with biologists and experimental researchers in order to refine. But the computation model is very important for this process because, as I said, we can actually do some sensitivity analysis. And if it turns out that some parameters have a big impact on the result, even small changes, then this is something we need to constrain further in collaboration with experimental researchers. And so this is an ongoing process.
Yeah, thank you. My other question is actually related. Can you more or less tell how many parameters you have for the cells and how many parameters you have for the surface when you do the modeling for biofilm?
Yeah, so there are different in silico models that we are using. I guess you're asking particularly for the biofilms, right?
Yeah, for the
Yeah.
biofilm.
Yeah. So for the biofilms, I mean, at the end of so many parameters that one could potentially set in a model, right? You could have different diffusion of the chemicals. You can have decay of the chemicals. You can have membrane viscoelastic properties of the membranes of the bacteria, and so on. So in reality, there are hundreds of parameters that potentially you can set. We usually work with around maybe 10 parameters in total that we try to, based on experiments, to fix. But yeah, I mean, the sky's the limit if you really want to go into as much detail as you wish. You can do that, but we usually, whether it's in biofilms or in cancer, we usually work with around 10 parameters, which is something that is reasonable with regards to the computational performance, because the more parameters we have, of course, we need to do also the more simulations we need to do in order to optimize the models, in order to validate with experimental data. But yeah, 10 is a reasonable kind of ballpark estimate.
Yeah, thank you very much. No, I was just curious because of course, if you need to do the simulation, you need to get the parameters in the lab experiment. I was just want to know more or less how many parameters you evaluate before. But thank you, really interesting work. I will give the floor for the other questions, but thank you again.
Okay, thank you. Stefan, you had a question.
Yeah, thanks for the talk. We are mainly working with artificial neural networks, so I'm interested in this simulation of neurons. And I was wondering if you had some insights in, like, first, if they would be trainable at all, but also if you would, if you got some insights also into maybe how to better design architectures of artificial neural networks, by simulating these cortical columns.
Very important question, and very interesting to me, because that's what the core focus of multiple PhD students is in my lab. So with regards to training, yes. So what Umar Abubakar is doing, I mentioned this cortical column with the neurite extension. He's growing them in Biodynamo, and then he's exporting those connectivities into Nest, and then he's actually training them and adapting the synaptic weights. So that's exactly where we are looking to go. At this stage, we are just trying to do some relatively simple computations, and Umar is currently writing up a manuscript. So once we have this a preprint, I'm happy to send you something along those lines. I had previously, like a long time ago, in 2014, 11 years ago, I have published a paper where we simulated in vitro growth. So this was not columnar growth, but it was like a 2-dimensional or 3-dimension growth of random growth. So this was not with layers. So this is more, let's say, more fundamental in terms of just having a neuronal network self-organize, and we could create neurons that then learn. So I simulated learning there. I can type in the chat the paper, and there we did not use spiking neurons. We used rate-based neurons there. And 1 second, I'm just typing it here.
And for the current architecture, you use spiking formulation of training? Or you plan to do that?
We currently use mainly spiking, but we recently submitted a paper where we also simulate multi-layer perceptrons, and there we used the rate-based models of neurons, multi-layer perceptrons, and we created sparse architectures. So that was a very good question about how can we use this insight to improve the architectures, because neural architecture search is a huge huge domain, right? And one of the problems there is that it's very computationally demanding to identify good architecture. So why not take inspiration from the brain, right? It's an obvious question to ask, and that's exactly what we did in that paper. There we created much sparser multi-layer perceptrons by taking inspiration from the brain. In particular, we looked at so-called topographic maps. So in the brain and also in the retina, you have topographically organized projections, and we kind of copied this idea, and we created multi-layer perceptrons that are, I think, around 96% more sparse than a fully connected network, and they sometimes have even better performance because it's not overfitting. And so this is something we are looking into that. We are currently also looking. I just had a meeting yesterday about this topic also. Anyway, if you're interested in collaborating along those lines, drop me an email and we can maybe discuss. Yeah.
Okay. Yeah. Thank you very much.
Okay. Thank you very much. Any other question? Well, actually, I had questions, but it was in the lines of Lennon and Stefan. So just maybe a comment. I was surprised to see the mouse brain has more the mouse cortical layers have more neurons than human cortical layer.
Yes. Yes. So the density is very high because they are smaller there, but that also depends on the where exactly you look in the brain, so which area you look at. But yeah, the density generally is higher than, for example, in human or monkey.
Okay. Thank you very much. Okay, so I hope we can keep in touch, as you said, on the biofilm simulation. And thank you very much again. Yeah.
Thank you. Yeah. Have a nice rest of your day. Oh, take care.
Yes, you too. Goodbye.
Thank you very much. Bye-bye.
Bye-bye.
Bye.